This tutorial will focus on another genetic tool called gel electrophoresis. It's a relatively simple idea, but a very, very powerful one. This particular genetic tool allows geneticists to separate DNA or proteins on the basis of their size. And the result of a gel electrophoresis is often a DNA profile or a DNA fingerprint. And this can tell us all sorts of different kinds of information. So let's have a quick look at the technique involved in gel electrophoresis. On the left here we can see an example of an actual setup of a gel electrophoresis tank. Essentially, let's start with the diagram on the right and work our way through how the generalized process works. So this is a tool, remember, that is going to be used to separate out DNA fragments according to their size or length. To create the DNA fragments from a sample in the first place, restriction enzymes need to be used. And if you're not sure about how these work, have a look at the tutorial on restriction enzymes. So if, for example, a sample from a crime scene, or it could be a sample from parents and children who have a disputed connection family-wise, and scientists are looking for similarities in the DNA of the two different samples, are going to use gel electrophoresis. So they may take the original sample, apply restriction enzymes to it, so the restriction enzymes chop up the DNA into chunks. Then a gel is prepared. Now the gel involves a tank, a plastic tank, and you can have a look at one of these in our own laboratory. Inside the tank is inserted a gel, a little bit like a jelly. The gel shown here is often made out of a compound called agarose. It's not unlike a compound like cellulose, but it's a mixture of agar and other kinds of sugars, and it's a porous kind of gel, not unlike a jelly, and it's set into a particular rectangular shape. And again, you can see this in our own laboratory. At one end of the gel are cut tiny little wells in which you can place the DNA. So what happens is that the DNA, after being cut by the restriction enzymes, as you can see here, are inserted into the various wells of the gel. And the gel then is placed into the plastic container, the plastic tank, and is covered by what's called a buffer solution. Now a buffer solution is basically a solution that allows electricity to flow through it easily. Now DNA is negatively charged because of the phosphate groups, so it's negatively charged. If you want DNA to move through the gel, you're going to have to apply a positive charge at this end of the tank and a negative charge at this end of the tank. You can see that over on this diagram here. So the black is the negative, and the wells will be situated along here, and the positive end of the electric circuit is down this end. And because DNA is negative, it will be repelled away from the negative end and attracted towards the positive end. So it will move this way through the gel. On this diagram, the DNA will move through the Agarose Gel this way. And that's essentially how gel electrophoresis works. Let's have a closer look at the Agarose Gel. In this diagram you can see a diagrammatic model of how the DNA can move through a porous gel like agarose. So you can see that the DNA fragments here are placed into the wells. So treat this as a single well in the gel. You've got the various, you've got the porous particles through the agarose compound itself, and the blue represents the DNA fragment strands. So they all start off in the well at the same place. Then as the electric current is applied, the DNA will move away from the negative end towards the positive end, and it's a relatively simple understanding to see that the smaller particles of DNA will be able to move further in the same time compared to the bigger particles, which will have greater difficulty moving through the pores of the gel. So over a set amount of time, say 30 minutes, of the gel running, the larger particles won't move as far as the smaller particles. And then at the end of it, we can apply various techniques to visualize the DNA and where it is. Since DNA is actually transparent, you won't be able to see it directly, but it's possible to either stain the DNA with various stains that can show up visually, and they would show up as DNA bands, hence a kind of a profile pattern. So the DNA found at this level, microscopically, would appear as a band, and the DNA that gets to this point in the gel will appear as this band here. And remember, the further the distance away from the original well, the smaller is the fragment, or the smaller are the fragment lengths. Other kinds of ways can be used to visualize. Let's have a quick look at some of those ways in the next slide. As we can see on this slide, there are various ways of visualizing the DNA bands that appear in a gel at the end of a gel electrophoresis run. In this one here, it looks like there's some kind of fluorescent dye that may have been attached to the DNA molecules that fluoresce in certain lights, and so kind of glow in the dark, so to speak. And that allows us to see the individual bands, as you can see here. You can also attach radioactive markers on the DNA, so add radioactive atoms to the DNA molecules, so that when you take a photograph, the radioactivity shows up. And this looks like something here, looks like a radioactive photograph. You can use simple stains like methylene blue, which you'll get to see in the laboratory. And methylene blue just stains or travels with the DNA, and you can see the DNA bands here, stained methylene blue, so a simple stain. At the end of it, though, you end up essentially with a DNA profile or DNA fingerprint, which allows you to compare DNA from different sources. And it's a general rule in gel electrophoresis work that if a band has travelled the same distance over the same time, then it has roughly the same length, or the same length as the other band. So if you had a look at this gel here, you would see that something like perhaps this band here and this band here have both travelled the same distance, so they're the same length. These bands here are all the same length. Remember, the further the DNA goes, the smaller are the fragments. So these fragments here, in this one, are larger than these fragments here. This is the smallest of the DNA fragments, that is the largest of the DNA fragments, and so on.